So our today's topic is fluorescence in situ hybridization. It is briefly named as FISH. So first of all, we will discuss its introduction. FISH is a technique which is used to detect specific DNA or RNA sequences in a sample by using some probe like fluorescent probe or radioactive probe. A labeled probe of DNA or RNA sequence is used to identify and locate specific sequence of nucleotides or genes in a chromosome present in the cells and tissues. तो एक ख़ास जो सीक्वेंस है डीएनए का या आरएनए का उसको यूज़ करते हुए हम अपने सैंपल के अंदर क्रोमोसोम के अंदर जो एक उसके कॉरस्पॉन्डिंग जो रीजन होता है जिसकी न्यूक्लियोटाइड सीक्वेंस उस प्रोब के न्यूक्लियोटाइड सीक्वेंस के साथ मैच करती है उसको हम डिटेक्ट करते हैं और जाहिर है इसको डिटेक्ट करने के लिए हम एक प्रोब यूज़ करते हैं जो फ्लोरेसेंट प्रोब होता है या रेडियोएक्टिवली लेबल्ड प्रोब होता है. विद दिस टेक्निक इट इज़ पॉसिबल to visualize and map genetic material to identify chromosomal aberrations, other changes or mutations present in genetic material of that organism. In situ term is used to study something in its natural position. As we study the specific sequence of nucleotides or we identify specific sequence of nucleotides in our sample in its natural position. That's why this technique is known as fluorescence in situ hybridization. I mean, we study the location of the gene in its natural position. This technique was first of all used by Gall and Padu in 1969 to identify a DNA sequence in the nucleus of a frog egg. This technique is used to detect the changes which otherwise are not visible in a microscope. We know when we see the cell in a microscope, we only see the bigger components, the larger components of the cells, for example, nucleus, cytoplasm, other changes. But we cannot see the sequence of the nucleotides. We cannot see the presence of the genes in the chromosomes. So that's why without this technique, it is not possible to locate or to identify those genes. But when we use some fluorescent probe, some other type of probe, we are able to see those genes with the help of these probes. The FISH is used to identify specific area or whole of the chromosome against which some probe is used. In case of some deletion in a chromosome, we don't see that part in the chromosome as it has been deleted. So we are able to detect only one bright spot instead of two because during deletion, one of the part of the chromosome may be deleted. So instead of seeing two spots, we see only one bright spot in the chromosome. And this technique is used to detect and localize specific RNA sequences even, but in the same way as we have discussed in the case of DNA. So हम DNA के साथ साथ जो RNA के sequences होते हैं उनको भी डिटेक्ट कर सकते हैं, मगर बेसिक प्रिंसिपल वही होगा कि हम एक रेडियोएक्टिव जो या फ्लोरेसेंट जो लेबल्ड प्रोब होगा वो यूज़ करेंगे और उसके कॉरस्पॉन्डिंग जो भी RNA का सीक्वेंस होगा जिसके साथ वो मैच करेगा उसको हम उसी तरह से डिटेक्ट कर सकते हैं.